Hi, Jerry. Good morning, everybody. Welcome to Grand Rounds. I see that people are still celebrating Memorial Day, but for those intrepid people who are here, I assure you that the Grand Rounds is going to be extremely interesting and worthwhile. First, let me mention that next week, our own Ted Huey. Ted, I saw walk in. Are you there? So you're up next week? And he will be talking about paradigm shifts in neurodegenerative disorders, implications for psychiatry. A couple of announcements. So Brett Kaufman, Anna Marcelin, Martin Picard received a 5-year R01 from SAMHSA. SAMHSA give R01s? Well, doesn't sound like their thing, but on transduction of psychological stress into systematic inflammation by mitochondrial DNA signaling. This sounds like it's a misprint. It must be the NIMH. And Harold Pincus was selected as the 2019 Mentor of the Year by the CTSA here, which is the Irving Institute Clinical Translational Research Unit. So congratulations, Harold. Also, at the recent APA meeting in San Francisco, of all the 700 or more posters that were presented, I think 5 were selected for media dissemination, and one of them was by our own Christine Hoven and Larry Amsel, and possibly others who were co-authors for their work on a 15-year follow-up of children of 9/11 survivors. So it was the effects of trauma on long-term outcome. Also, there will be in your email today a email from President Bollinger announcing that Dean Goldman will be stepping down at the end of this next academic year. That'll be the conclusion of his 3rd term as dean, and he's chosen to step down, and having presided in that position for the last 14 years. So presumably there'll be a search thereafter. So the details will be forthcoming today, but I figured I would tell people in advance. So it's really a pleasure to have Jerry Dawson back with us. So Jerry was scheduled early this year, but weather or something intervened, and we weren't going to let her get away, so here she is. But Jerry, are you from the West Coast originally? She's from the Pacific Northwest. Did you ever read that book? What is this? Alice doesn't live here anymore? No, that's not it.
I've heard of it. Wasn't that sort of an early generation?
No. I'm misremembering it. I need to either. You're dating me, Jerry? Okay, well, it serves me right. At any rate, she hails from the Pacific Northwest, and she went to undergraduate school at the UW. She received her PhD in psychology, developmental psychology, at UW with her dissertation on early infantile autism and hemispheric specialization. And then she did a postdoctoral fellowship at UCLA in neurodevelopmental disorders. Subsequently, she went to North Carolina. Now, you might say, well, why is somebody from the Pacific Northwest going to North Carolina? And because at that time, which was 1980, autism was barely on the map in terms of a condition that was the focus of academic medicine. And for reasons that I'm not entirely clear about, even though I should know having been there for 10 years, the only or the most developed program to address the needs of families with autistic children was in North Treatment It was called TEACH. That was the acronym. Do you remember what it stands for? Treatment and Education for Autistic and Communication Handicapped Children. And this was something that people all over the country would move to North Carolina for because there was nothing for them. You remember that autism didn't appear in the medical literature until the 1940s with Leo Kanner, and then nobody took it seriously, and really until probably the late 80s, early 90s, when famous, prominent people's children or grandchildren started developing it and finding out there was nothing for them. So Jerry went to North Carolina. She was an assistant professor there until 1985 when she was recruited back to UW, where she remained for a good number of years, ascending from assistant to professor. In the course of that, she directed the Child Clinical Psychology Program, the Autism Clinical Program, the Autism Spectrum Disorders Clinic. She was co-director of the Integrated Brain Imaging Center, directed the Brain Science Behavioral Core of the Center on Human Development and Disability. She was the director of the NIH Collaborative Program of Excellence in Autism, and so on and so on. She remained there until 2008 or so, when she was lured by some people that we know very well, the Slatkins, the Wrights, the Simons, and so forth, to come to New York to be the scientific director of Autism Speaks, which was the largest and most influential advocacy organization in the country. And there she really played a very different role as being kind of a visible public spokesperson, an advocate for research and clinical care, and also somebody who was a source of guidance and information in terms of policy for the federal government. While she was there, she maintained an academic affiliation with the Department of Psychiatry at the University of North Carolina. But then in 2013, she stepped down from her position at Autism Speaks. She returned to North Carolina at Duke, where she currently is professor in the Departments of Psychiatry, Behavioral Science, Pediatric Psychology, Neuroscience, director of the Duke Center for Autism and Brain Development. I don't know what sort of initially led Jerry's interest in psychology and child psychology to autism, but she certainly was there kind of at the beginning, at the right time, and I think she, as much or maybe more than any other person, kind of personifies the serious scientific effort to try and understand something which was basically a black box and was unknown. You know, before the movie The Rain Man came, the public had little, sort of visible awareness of what autism was. And for many people, that was, Dustin Hoffman was like their first impression of what autism was as a condition that affected humankind. But Jerry, by dint of interest, prescience, and sort of luck, was there at the beginning and has been a steward of the development of the field over its really most productive periods in capacities as researcher, clinician, policymaker, and now stewards this program at Duke in North Carolina, which is where it all began, although the approach that Teach took then was not something that would be considered state-of-the-art at this point. In any event, I could go on, but I won't, other than to say that she's a fantastic scientist, great clinician, wonderful person, and the only thing I could say it would be better is if she were at Columbia here in New York, but we'll work on that, Jeremy. But please join me in welcoming Dr. Jerry Dawson.
Well, thank you for that wonderful introduction, and actually it's very appropriate because my talk is going to be looking back and sort of taking us through that history that you just alluded to. And I will mention that how I originally got interested in autism as an academic. I actually had exposure as a younger person as well, but in my training, my first case was a child with autism at the University of Washington, and it was so rare to see a child with autism that we actually had a grand rounds about this child. And Bob Reichler was there. He arranged this this grand rounds, and they flew Eric Schopler from University of North Carolina in Chapel Hill all the way to Seattle to be part of this grand round. So my introduction was standing on the stage with Eric Schopler talking about my first case as a clinical psychologist who was this child with autism. And at the time, we really knew nothing about why is it that a child would come into the world and not be able to form social relationships? Because we didn't have concepts that we take for granted right now in terms of social cognition. So Mike Rutter, I remember, had a very influential article where he introduced the idea that of social cognition, which is something that we just, we all take for granted. We knew very little about the neurocircuitry that was responsible for social behavior, except for the limbic system that had to do with motivation and things like that. So it was a real mystery, and I knew that intellectually to understand that would teach us something very fundamental about ourselves as human beings, but also at the same time we had very little knowledge around treatment. So I had nothing that I could offer this wonderful family and child that, except for, what they were starting to do in the University of North Carolina, Chapel Hill, which was a parent-delivered treatment, which was, of course, now these are very common. So anyway, thank you for that introduction. I also want to thank Jeremy for the invitation. It's wonderful to be here. I also want to give my regards from Maura Wren. Sorry we stole her from you, but we are very much benefiting from the wonderful experience that she had here, and she's brought a lot of that with her. So today I am going to be talking about paradigm shifts, and I'm going to start by looking back and describing some of the fundamental paradigm shifts that shaped our thinking about autism. And then I'll end by looking forward at what I think are the paradigm shifts that are just currently emerging, and I think will have a large influence on autism research going forward. Here are my disclosures. The first paradigm shift was the concept that the autism symptoms that we see and diagnose are perhaps the secondary consequences of early differences in the way that the infant is paying attention to the world. So what we learned early on is that the brain is developing rapidly during the first several years of life. And for the brain systems that underlie social and language behavior, we need to have the appropriate kind of input, otherwise they're not going to be developing normally. And so if an infant beginning, say, 6 to 12 months of age is not paying attention to the social world, this means that the baby is not getting the normal kind of stimulation to those neural systems that underlie social and language behavior. And so what we might be seeing by the time that we diagnose autism, even at 18 months or sometimes 3 to 4 years of age, is really the secondary consequences of these early differences in not only the way the baby is paying attention to the social world, but their affective engagement. So those are two key concepts and things that are different very early on, even before the full syndrome is manifest. William James said, "My experience is what I attend to, and only those items I notice shape my mind." So what we begin to pay attention to in our lives is going to shape not only our minds, but the neurocircuitry that supports our thinking and being. So that, I think, was a very key concept in understanding the full array of symptoms that we think of as autism spectrum disorder. Now, in the early 1990s, we published, first of all, the first case study of an infant that was diagnosed with autism at 13 months. This was a very interesting case study where we had a detailed record of that baby's development from birth by a neurologist who had kept track of the development. And that neurologist described between around 6 to 12 months that the baby became less interested in people, became more interested in objects, And then by 13 months, this particular baby had the full array of autism symptoms, which include difficulties in social interaction and the presence of repetitive behaviors in a restricted range of activities. Then we also decided to use home videotapes to understand early development, because at that time, people thought that autism probably couldn't be diagnosed until age 3. They felt that you needed to have deficits in language and also imaginary play, and these are things that don't emerge till age 2 or 3. But in thinking about the symptoms of autism, which is poor eye contact, for example, we knew that these are things that are there in infants. And so at the time, we decided to use home videotapes to look back to see whether we could see some of these symptoms. And here, what I'm showing you is a typical baby at their 1-year birthday party. And in this case, the baby as people around the baby are singing happy birthday, they're bringing in a cake, which has a flaming, candle on it. And I always think about, what IRB would approve you bringing in a flaming object and putting it in front of the baby, which is a traditional, what we call social referencing experiment, where you put something novel in front of the baby they've never seen before. And what babies typically do is they look at the people around them, and they use the responses of the people around them to know how they should respond. And this also implies that the baby is interpreting and understanding the emotional expressions of the people around. So you can see this baby's very interested, is affectively engaged and listening to the singing. Whereas the 1-year-old with autism is very focused on the plate. You also see the flat affect and the lack of affective engagement with people. So we now know by using studies of high-risk infants, so infant siblings who have an older sibling with autism that we follow longitudinally, we know that these symptoms can be verified prospectively in these longitudinal studies. Now, as I said, at the time we also knew very little about social brain circuitry and what was the reason why children were not interested in faces. And but in the 1990s, particularly when people started using fMRI, people like Nancy Canwisher started to describe the neurocircuitry that is activated when you or I or an infant looks at another person's face. And what she and others described was a complex neurocircuitry that included the fusiform gyrus, which is responsible for detecting the identity of the face. Also, the superior temporal sulcus, which is responsible for interpreting facial expressions. This then, interfaces with areas of the brain that are responsible for speech, such as the anterior temporal cortex, and these are also linked to the amygdala, which is responsible for the emotional response to a face. So this complex neurocircuitry is activated when a baby is looking at their caregiver's face. We also can interrogate this system in very young infants by using event-related brain potentials. So EEG has allowed us to study early brain function from a very, very, very young age. This can be used with severely affected children who don't have language. It can also be used with neonates and infants to study brain development. And you can see here in this graph that the affective or attentional engagement actually modulates the amplitude of the ERP signal. This is the later signal or later components of the ERP signal when a baby is looking at faces or toys. And these are periods that were defined by heart rate that indicated that the baby was affectively and attentionally engaged with the stimulus. So the brain becomes more activated, and you can measure this with the amplitude of the ERP signal. Now, in the early 2000s, we began to publish studies to show that this system was not functioning properly in young children with autism. So we conducted a number of studies with toddler and preschool-age children with autism, comparing them to typical toddlers who did not have autism. And in this study, we showed these young children, first of all, a picture of a stranger's face, and then a picture of their mother's face, and then a picture of a favorite toy versus an unfamiliar toy. And what we found is that 3-year-olds with autism had a very nice and robust differential ERP to a familiar versus unfamiliar object, but they showed no differential ERP to their mother's face versus a stranger's face. Now, keep in mind that it had been shown, people like Chuck Nelson had shown that by 6 to 7 months of age, that if you show a baby a familiar face versus an unfamiliar face, you get a robust differential ERP response. So this meant that systems that should be online by 6 months of age, that are part of the very basic social brain circuitry, are not developing normally in children with autism. We were also able to show that not only do we see differences in face identity, but also in response to emotional faces. So a fear face, for example, is well known to activate the amygdala as well as other aspects of the face processing system. And so we were interested in trying to understand why children with autism seemed less sensitive in reacting to emotional cues of other people. And actually here is an experiment that we did where the child was just playing with toys, and then in the middle of the play, the experimenter, who happens to be Jamie McPartland, in this picture, when he was my graduate student. So this was when he was earning his PhD with me at the University of Washington. He's now head of the autism program at Yale. But he would hit his finger and start crying. And we were interested in how quickly the baby, or the child in this case, looked up and noticed, as well as how long they sustained their attention. And then we brought them into the lab on another day, and we measured the ERP to a fear face. And what you see here is that typical toddlers and preschool-age children show a robust, what's called a P300, a positive going component at about 300 milliseconds, as well as N300. Whereas the children with autism did not show this P300 response in the same robust way. Furthermore, the latency of that P300 response was highly correlated with the amount of attention they had spent looking at that distressed experimenter on a separate day. So here you can see the correlation where if your brain is responding very quickly to a fear face, you're also a child who pays longer and more robust attention to a person who's distressed on a separate day. So we started to see that these brain measures actually had some ecological validity in explaining some of the symptoms of autism. Now, to try to understand these findings, we also proposed something called the social motivation hypothesis. And the idea here was that perhaps the underlying deficit in autism has to do with the sensitivity to the reward that is associated with people. So for the typical child, you don't have to teach them to be interested in faces. Faces are wired into the brain as a reinforcing stimulus, right? They're a primary reinforcer. Whereas for children with autism, we speculated or hypothesized that the systems that underlie this natural motivation to want to look at faces that is there during the neonatal period is deficient. And this may have to do with oxytocin or other aspects of reward circuitry. So then this leads to less attention to social information, faces, language, gestures, and that this then would lead to a failure to develop expertise in face processing, and also a failure for those parts of the brain that are responsible for social processing to become organized, specialized, and to function efficiently. And you can see this when we map this component called the N170, which is a face-specific component. So the N170 is very robust in whenever a face is shown. You see a faster N170 as well as a greater amplitude of the N170 response. It's a negative going peak in the event-related potential that's about 170 milliseconds after you show a face. And when we map this across the scalp, the topography, you can see that in typical children, it shows a very nice right lateralized response, which is what we'd expect, whereas in the children with autism, you see that the pattern is very diffuse. So it suggests that these systems are not becoming tuned up or specialized with development, we believe, because they're not getting the normal kind of input from the environment. Now, this has implications for thinking about mediators of treatment. So the idea here is that if the child has reduced sensitivity to social reward, and this then leads to a lack of interest in people, that these may be targets for interventions. You want to draw the child's attention to the social information. And it is these very early differences in the brain then that become mediators for opportunities for social learning. So you're not going to develop social skills and language if you're not paying attention to them. And then this would then eventually lead to our clinical inputs in a clinical trial, such as the acquisition of social and language skills. So you might expect, for example, if you were testing a medication that you thought improved social brain circuitry, that you would first see early changes in these attentional systems or even in aspects of, say, the N170 response. But this would signal that the attentional and neurosystems are changing, and that then through interaction with the world, the child would start to learn and gain these social and language skills, and those would be endpoints that would be farther in time to develop. So people have then become interested in whether we can use social attention, which we can measure both through eye tracking, what are you paying attention to in the world, or you can measure with the N170, how fast is your brain responding to a face, for example. We're now using these as biomarkers in clinical trials, and they can be used also for early risk detection. I'll show you some data on that in a minute, for stratifying children entering into a clinical trial, or, as I said before, as an indicator of early efficacy in a clinical trial. And in fact, now NIH has invested about $28 million in a consortium that's called the NIH Autism Biomarkers Consortium for Clinical Trials. This is headed up by Jamie McPartland, so it's a wonderful story, and from a graduate student testing the N170 in my lab at the University of Washington to now heading up a multi-site trial. So he's the overall PI in a trial that involves Duke. So he's now my boss, which is great, and it involves Yale and Harvard, University of Washington, and UCLA. And what we've been doing is conducting a multi-site naturalistic study that is following, it's now over 200 children with autism between 6 and 11 years of age, following them longitudinally, as well as a sample of typically developing children at the same age. And we're looking at a wide range of both eye tracking as well as event-related potential and other EEG biomarkers, with the idea of validating those for clinical trials. And most of these are tapped into this concept of social attention. So we see the kids at baseline, then 6 weeks later to look at test-retest reliability, and then 24 weeks later, and following the kids clinically to see whether any changes in clinical outcome track with changes in these eye tracking and EEG biomarkers. So we now have 2 biomarkers that have passed a threshold for reliability and construct validity, test-retest reliability, And they have been submitted to the FDA, and we have gotten feedback from the FDA that the N170 to faces is likely to be the first biomarker that will be validated for use in clinical trials, particularly as a stratification biomarker for entering into a clinical trial. So it's exciting to see that we moved from an initial concept to now perhaps an FDA-approved biomarker. So a corollary of this paradigm shift is that if the way that we pay attention to the world can influence brain development, that we should be able to develop interventions that change the way that the child is engaging, both in terms of their attention and their affective response. And this could influence the trajectory of both behavioral and brain development. So my colleague and I, Sally Rogers, developed an intervention that is part of a broad group of interventions that we now call naturalistic developmental behavioral interventions. So these really grew out of traditional ABA interventions, but they have a strong focus on naturalistic strategies, a very strong focus on the relationship quality, because we know that affective exchange is very important. We do use applied behavior analysis strategies, but they're ones where we use children's preferred activity. We have a strong focus on the child's motivation. It's very sensitive to normal developmental sequence in terms of the objectives that are targeted, and there's deep involvement with parents, because we know that every interaction is an opportunity for learning, so we want parents to be able to use these strategies at home. So the Early Start Denver model, as I said, is one example of these NDBIs, as they're so called now. It's been translated into 17 languages and is used worldwide. Now, our first randomized clinical trial was published in 2010, and it was a study where the children were randomized either to receive 2 years of the ESDM intervention. This was in the children's home, where we trained therapists to go and work one-on-one with the child at least 15 hours a week for 2 years. And we compared this to treatment as usual in the community. We found a number of improvements in IQ and language and adaptive behavior. And then when we looked at these carefully, what we found was that the improvements in IQ were largely driven by both receptive and expressive language gains. So you can see here when we looked at the two groups and looked for a group by time interaction, that both receptive and expressive language showed robust improvements, both at 1 and 2 years, but the areas of nonverbal IQ, there really wasn't a difference between the community group and the ESDM group. So we then decided to look to see whether we could document any changes in brain activity, kind of, again, trying to see whether there could be an impact of this early experience on brain development. We used the same kind of paradigms that we did before, where we showed familiar faces and familiar objects, and then unfamiliar faces and unfamiliar objects. And we took a number of brain measures. We did look at the N170, and we found that the later components of the ERP, those that I showed you very early in this talk that are sensitive to attentional engagement, that there was differences between the children who had received ESDM, where now the children who had received ESDM showed a more robust amplitude of the later components, suggesting there was more attentional engagement to the faces. We also did what's called spectral analysis of the ERP signal. So this allows us to break down the ERP into the different spectral components, and some of these are connected with active information processing. So for example, alpha decreases when the brain is actively engaged, and theta increases. And what we found, and you can see here, is that the children, so we also brought in a group of typically developing children. So these are kids, all kids who are age 4. They have now received 2 years of intervention or been followed in the community for 2 years. And you can see from this pattern, and I won't go into all the details, but essentially this pattern is saying that the brain is much more activated when you're looking at objects than you're looking at faces, which is what we had shown in our early studies. But now the children in the ESDM group showed a pattern of brain activity that was really indistinguishable from a typical child. So now they showed more brain activation when they were looking at faces than objects. So this was the first study to demonstrate that early intervention could have an impact on brain activity. Now, children went their merry way, but we followed them and we chronicled what kinds of services the parents were seeking out for their children. We were very interested in whether, if you received that early intervention, did it have any impact later in terms of the utilization of services, and then could you see any long-term gains in terms of things like IQ and autism symptoms and so forth. So one of the things that we discovered is that children who had received the 2 years of intervention now sought out far fewer services. In fact, David Mandel conducted an economic analysis of the cost of each one of those services that could range from speech therapy to one-on-one ABA therapy and so forth. And then he was able to calculate how much savings the children who had received early intervention were having, because they were now seeking out fewer services. And this has been published, and what he found was that the annual cost savings was about $19,000 a year in terms of reduced use of services in the children who had received 2 years of intervention. We also did find stable long-term gains in this group. So you can see, comparing now the children in the community group in green versus the ESDM group, in green, and the community group in blue, that we see continued differences in IQ over time. We also saw a drop in the ADOS severity, which is a measure of the severity of autism symptoms that was significantly different at age 6. So this suggested that early intervention did have a sustained effect later on. Now, just recently, we have published a multi-site randomized clinical trial comparing ESDM to community intervention. To my knowledge, this is the first single-blind, randomized multi-site trial of a comprehensive intervention. These are very hard to do because you're seeing kids for 2 years, 15 hours a week, and maintaining fidelity of those therapists doing that intervention over that period of time. 118 children, 14 to 24 months. It began as 3 weeks of parent coaching and then 24 months of 15 hours a week of one-on-one treatment. Children were randomized either to the ESDM group or to the community group. All the evaluations were conducted by people who had no idea which of group assignment. In fact, it was done in a completely different building. And because we had noted in the earlier trial that most of the gains were in language, we chose as our primary endpoint language age equivalent on the Mullen scales of early learning. Now, this is a very high threshold, so many of the early intervention studies for their outcomes are choosing what we call proximal outcomes, where we look at parent-child interaction, parent-child synchrony, whereas having a child perform on a standardized test by someone outside, the parent-child context is a much higher bar. We did find that the ESDM group performed better in terms of language gains compared to the community group, so we were able to validate improvement on our primary endpoint. Interestingly, if we looked at overall IQ and adaptive behavior, there was no difference between the groups. And what we saw was that the community group now, in contrast to the 2010 study, had also made large gains. So the amount of gains that we saw in IQ and adaptive behavior in this study in the children who had received ESDM was equivalent to what we found in the 2010 study. The difference was that the children in the community group now also had made those gains. And we can speculate why that might be the case. I think, a large part of these kids were from California and Seattle, where they have access to many services. So I do think the kids are getting much more service in the community group now. And in fact, they had the equivalent number of hours of intervention. All right, so we're going to move now to the second paradigm shift, which is a shift from the idea that we want to diagnose autism as early as possible and then provide these treatments with the hope that we can alter both behavioral and brain outcomes, to the idea that could we detect autism even before it's fully manifest and begin interventions during that prodromal period. And what we want to do here is to really intervene before the period of diagnosis with the goal of even reducing this gap further. Now, this is a very active area of research. There are many, many studies that are now looking at different kinds of early bio and behavioral markers that could indicate that a baby is at risk for autism. Some of these are looking at attention, vocalizations, eye tracking. There's work using EEG and MEG and NIRS. There's a wonderful study that Joe Piven is heading up where he's following large groups of infants who are at risk for autism by virtue of having an older sibling with autism, and then looking at MRI longitudinally and seeing changes by 6 months of age that are different in terms of the brain structure development. And of course, there are people looking at blood-based biomarkers in terms of genome sequencing, gene expression profiles, microRNA, and other indicators from the blood that could suggest that this baby is at risk for autism. So obviously, I'm not going to give you a review of all of that. I'll just give you a little taste of the kind of work that is coming out of this, showing you some work from our own lab. Now, as you've seen, we're very interested in using EEG as an early biomarker. And so we recently published a study where these are 6-month-olds all of whom have an older sibling with autism. And so at 6 months, we used that same paradigm of presenting faces and objects and looked at the ERP signal to a face to see whether that might predict which baby would go on to develop autism. And we did find significant differences in the ERP to a face at 6 months, which went on to predict those children who developed autism. So then the question is, can we present an intervention during this period where the baby has not yet developed symptoms and stimulate early brain development to protect, in a way, the baby from going on to develop autism? So we've only published part of this study, so I'll show you the first part today. But the way in which we conducted the study is, again, a group of 6-month infants. This was a proof of concept study, only 33 babies, all of whom had an older sibling with autism. That means they have about a 1 in 5 chance of developing autism. They were randomized either to receive an intervention that involved teaching parents to be sensitive to the baby's cues and to provide stimulation, both social interaction and linguistic stimulation for the baby, or they received just no care because you don't really treat babies at this age. I want to point out that these babies were not necessarily symptomatic, right? So there is a published study, I don't have time to go into the details, where they picked out infants who were showing symptoms and did a similar kind of approach. In this one, it was just you're at high risk for autism. Whether or not you have symptoms or not, you'll be randomized to receive intervention or not. Then at both 12 and 18 months, this study was to look at whether we could alter some of those underlying constructs that we were interested in. So we had a paradigm where we looked at the baby's ability to encode and recall faces using a habituation paradigm. And then again, we used EEG, both our ERPs to faces that you're familiar with by now, as well as we looked at spectral analysis while the babies watched dynamic movies of social and non-social stimuli. So what we found was that this low-intensity parent-delivered intervention did positively influence both encoding and memory for faces. So what we found is that at 6 months, there was no difference between the two groups, whereas by 12 and 18 months, the babies who had received the interventions now were more rapidly encoding faces. Now, interestingly, there was no difference at any of the ages in their ability to habituate to objects. So this is very specific to face memory. We also found that there was a significantly greater frontal power, theta power, at 12 months in the babies who had received intervention. And then also the ERPs now were more similar to a large group of typically developing babies that we also assessed in this study, both at 12 and 18 months. So this was a first proof of concept that providing these low-level parent-delivered interventions could alter some of these fundamental aspects of early brain development that we know are awry in autism. And we're now looking at the clinical outcomes in this sample. Okay, so now I'm going to shift fairly dramatically, because one of the things that we learned from doing these early intervention studies is that whereas children benefit substantially from these interventions, there's still a large group of infants or children who have poor outcomes. So about 30% of individuals with autism do not develop language, for example. And so there is a need for medications, and both molecular and cellular treatments, that could promote neuroplasticity and help those individuals who need more than a behavioral intervention to have a positive outcome. So as I left Autism Speaks and went back to Duke, this was a primary focus that I wanted to be involved in, is testing medications that might be able to enhance neuroplasticity, with the idea that we could combine these with behavioral intervention for those that haven't had a positive outcome. So this is the next paradigm shift, which is that we could actually develop medications that could address core symptoms. Keep in mind, there are no FDA drugs that can treat core symptoms of autism, such as their social and communication abilities. Only drugs that treat associated symptoms, such as irritability. So currently at Duke, we're conducting a number of trials. I'm not going to go through all of these, that are focused on testing medications where the target symptom is either joint attention or social communication skills. These are focused on different mechanisms. Some of these are looking at reward circuitry, such as oxytocin or vasopressin, dopamine, and then we're conducting a large program of research funded by the Marcus Foundation that is looking at potential cellular therapies. And I'm going to just walk you through an example of an open-label trial testing these cellular therapies. Not because we have evidence yet that they work, but I want to show you how we can incorporate some of those assessments and biomarkers that I've been discussing into our clinical trials and do a better job of testing the outcomes. So I'm going to quickly tell you about our work in the area of looking at cellular therapies for autism, beginning with an open-label trial that evaluated autologous or a child's own cord blood as a potential treatment. This work is being done in collaboration with Joanne Kurtzberg, who is a pediatric oncologist at Duke and a world leader in the area of cord blood and cellular therapies. So as you may know, there is some evidence for neuroinflammation in autism. So in postmortem studies, we see different gene expression profiles that indicate microglial upregulation. There's inflammatory markers that you can see in neonatal blood spots that are associated with later risk for autism. There was one study, a PET study, that showed increased microglial activation in the brains of adults with autism. I should point out that that study has recently been critiqued, and I don't know if it will be replicated, but it certainly motivated our work at the time that we began this work.
What's the ligand?
TSO. Yeah, and that is actually the criticism, that it's nonspecific necessarily to microglia. So I can get into that in terms of a failure to replicate that I literally found out like 4 days ago. So, but yeah, I mean, I think it was interesting, but not the only reason we were motivated to look at cord blood. So what we know is that in animal studies that infusion of umbilical cord blood does reduce microglial activation. Those have been published. It also stimulates repair of myelin in both human and animal studies. There's been a double-blind randomized placebo-controlled trial in children with CP where autologous umbilical cord blood improved motor function in children with CP. And the idea here that in terms of mechanism is not that these are stem cells that are engrafting in the brain and creating new neurons, as you would in other applications of cord blood, but rather that it's a cell-to-cell signaling mechanism that has an anti-inflammatory or immune modulatory mechanism, perhaps release of neurotrophic factors that improve brain function. So our first trial was 25 children, 2 to 6 years of age. We had as our primary endpoint the socialization subscale on the Vineland, which is often used in clinical trials these days. We did see a significant increase in the mean Vineland socialization score. We also noted that children with higher IQ made greater increases. We also found that the increase was not correlated with the number of hours of behavioral intervention. Now, we had a number of secondary endpoints, so the Clinical Global Impression Scale, the Pervasive Developmental Disorder Behavior Inventory, and in a standardized, administered by a clinician, expressive one-word vocabulary test. And in all of these, we showed improvement. Now, of course, that doesn't mean that there was we can't attribute this to the cord blood at all. It could be that children are just getting better, particularly on the parent report measures, because we know that there's a very significant placebo effect in autism. We did use eye tracking, so this social attention, how much attention are you paying to social stimulus in a complex dynamic social stimulus. We found that at baseline, these eye tracking measures of social attention were highly correlated with our clinical endpoints on the Vineland. And then we also showed that a 7-point increase on the Vineland was associated with a 14% increase in looking at the actress and eye tracking. So that was better. I mean, still doesn't say that cord blood was the reason, but now at least we had an objective biomarker. We also used EEG to look at changes in brain activity. We were very interested in this finding that in autism you see lower levels of power in the mid-range frequencies. This is thought to reflect this GABAergic tone or altered inhibitory excitatory balance, where when you see reductions in alpha, this is an excited brain, so to speak. What we did find was significant increases in frontal and central alpha and posterior beta EEG power, as well as significant decreases in posterior theta power. So we did see changes in brain activity. Again, could have been development, but it was encouraging. Children also had an MRI, both at baseline and at the 6 and 12-month outcome in this study. And then with Alan Song and Kim Carpenter, we used deterministic tractography to look at the connections between brain regions where we could actually measure the number of fibers connecting different brain regions. And we did see greater connections in areas that we consider social brain circuitry, such as the fusiform and the superior temporal sulcus. And these were correlated with some of the outcomes, including our primary endpoint of socialization on the Vineland. So this was all very encouraging. And so we have just completed, and in fact completed the data analysis, but we're not able to share that with you today, but we hope to release those findings this summer. But this was a phase 2 crossover randomized placebo-controlled trial. Children were randomized to receive placebo, where they receive an infusion that looks and smells, and you cannot tell the difference between that and receiving an infusion of cord blood, or the other arm was randomized to receive cells. If they had their own cells, they received autologous cord blood. If they did not, they received allogeneic or donor's cord blood. This was 180 children who were seen at both baseline and 6 months and then followed remotely at 12 months. They were 2 to 7 years of age, and as I said, we have just completed analysis of those data, and I'll be able to share more with you this summer. But based on that, I will say we're moving forward with another large trial that will be actually testing mesenchymal stromal cells, which are made from cord tissue. The purpose being that we can do multiple doses from one tissue, which you cannot do with a sample of umbilical cord blood, because you usually have only one unit of and one dose. So that study will begin this summer. All right, I'm going to quickly end by telling you just the last paradigm shift that I'm excited about. And as you've seen, one of the things that I'm very interested in doing as a psychologist is being able to provide good measurement in clinical trials that are testing these medications, whether they're cellular or molecular medications. And we have a problem in the field of autism that when we do these trials, we often cannot tell whether the medication did not work or we just don't have a good assay of change. And this is true particularly when we look at behavior. So when we're using parent report, for example, there's such a huge placebo effect, and we certainly saw it in our trials, that it's very hard to know whether you're really seeing change. And so we need better measures of behavior. And this leads us to the last paradigm shift, which is the impact of digital phenotyping on our ability to detect, diagnose, and assess outcomes in autism. So I think that we have a major challenge that's associated with clinical ratings and parent report. These have poor resolution. They're based only on the clinician's naked eye. It requires a high degree of training often to maintain reliability. We have this influence of expectancy or placebo on the ratings. For many populations, our questionnaires present a literacy or language barrier. We now know that these can be highly influenced by parents' knowledge not only of their own child, but of child development. And also, it's very hard in many cases to collect very large data sets for some of our newer analytic techniques because of the effort that's involved in either doing behavioral coding or some of the more complex clinical ratings that require reliability training. I believe that digital assessments can potentially address these challenges by providing higher resolution, capturing the dynamic nature of behavior. They're automatic, objective, reliable, scalable. You can export them to natural environments, and I think this will allow us to collect those large data sets and use techniques such as machine learning to be able to have improvements in our measurement. So our first proof of concept study was with 104 toddlers. This was done out in Duke Primary Care, where toddlers watched a set of movies that elicited different behavioral responses. The camera in the iPad then recorded the child's behaviors, and we were then able to look at a wide range of behaviors that were coded through computer vision analysis. 22 of these toddlers were diagnosed with autism. And so one of the things that we did was during the assessment, the examiner called the child's name. We were interested in whether we could measure orienting to name, which is a key early autism symptom. And so you can see the computer telling us when the child is orienting down here. And this is the correlation between human coding of the orienting behavior and computer coding. So we're very good with the computer at being able to measure orienting to name. Now, we've called the child's name 3 times while the movies were shown. children with autism were less likely to orient. That's a well-established finding. No child oriented all 3 times if they had autism. But here's the key point, is that some children with autism did orient, and what we found was that on average, the latency to their orient was about 1 second slower than the typical kids. This is not something a clinician would see with their naked eye, and they would have indicated that that child does not have problems with orienting. So again, this fine resolution is going to give us better measures of behavior. It also leads to novel discoveries. So one of the things that we discovered is that when young children with autism watch a video, they have a subtle head movement throughout that video. And we saw this across all of the videos. As you can see, the robust differences between the non-ASD and the ASD children in just the frequency of head movements while they're watching these different videos. And I'll just show you what this looks like here. So this is a child without autism watching this little bunny video. It sped it up so it doesn't take too long to show you. Now let's see what a child with autism looks like watching that same video. And we're just looking at the cumulative number of head movements that can be detected through computer vision analysis. We also can see differences in affect. So we found that toddlers with autism display more frequent neutral or flat affect, which we can easily measure. So we see on the top here, the computer is telling us the probability that the child is displaying neutral affect, and its red children are children with autism as compared to the typical children. Also, you can see the difference in variability. These are the odds ratio of those intervals of neutral affect, and you can see that there's a significant difference between autism and non-autism. Another example is that when typical children watch an engaging movie, their face brightens, and this can be measured with the widening of the eye and the raising of the eyebrows. We call this engaged interest, and we saw robust differences between toddlers with autism and typical toddlers in this engaged interest. So here I've plotted the computer is telling us second by second the probability that the children are expressing engaged interest, and you can see that the non-autism toddlers in blue are showing this engaged affect, and the children with autism are not. Again, these are things that clinically we probably detect, but we have not been able to measure. Now, one of the great things about this approach is it's exportable. We conducted a study where parents were able to download these movies on their iPhone at home from iTunes, and this allowed us to collect 1,756 children over a year period. And this is what I mean about scale and being able to get large data sets from community samples that don't require the parent to come into a clinic. And we were able to show that you can get good data by parents showing on their iPhone movies and having the camera in the iPhone be able to record the child's behavior. And we showed significant differences between children who are at risk for autism by parent report on an MChat versus children at low risk in terms of their patterns of affective expression as well as patterns of attention. So we now have completely redesigned this app based on new technological advances that the Guillermo Shapiro and his team at Duke are now capable of. And we now can measure multiple autism risk behaviors. So several behaviors in the area of attention, facial expression, postural sway. We also are seeing differences in the vocalization patterns of children as they watch these movies. And then we have developed a couple of games that involve bubble popping, where we're looking at a variety of motor activities, including speed, accuracy, force, and repetitive behaviors, all either on an iPhone or iPad. We're actually testing both modalities. Our current research, we have received an NIH Autism Center of Excellence Award. The award is focused on understanding the comorbidity between autism and ADHD. And one of the projects in that ACE Center Award is inviting every child who comes in through 4 Duke primary care clinics to participate in our study as they come in for their 18-month well baby checkup. Now at Duke, they all get the modified checklist for autism in toddlers, which is the parent questionnaire that is standard for autism screening at 18 months. But they're also receiving our app where we're showing a variety of movies to see whether we can do a better job at being able to detect not only autism, but distinguish it from related neurodevelopmental disorders such as ADHD. So I want to end by just thanking all of my collaborators over the years. It's so many different people from so many different disciplines have made this work possible. And I also want to thank all the funding agencies. And I'll end by showing you this beautiful painting. And this is of Duke Chapel, painted by an autistic adult who's Jesse Park, one of the early artists who have autism. And so I hope you enjoy looking at this painting. Thank you.
Jerry, thanks for this presentation, but it's just an astonishing body of work that you've been able to accomplish with colleagues. Really, really impressive. I mean, this is exemplary. I don't want to be negative or critical.
Bring it on.
This is New York. And I'm a Tar Heel from UNC and Blue Devil.
Oh, okay, now the truth comes out. He's a Tar Heel, all right. No.
Well, we missed our chance to get Zion Williams, so.
That's true.
But the work that you've done is as elegant and as rigorous and as thorough as one could try and address a very complex and mysterious ailment. Do you feel like you have gotten traction on this, or you're just sort of barely scratched the surface? And what I mean by that is that in terms of the biomarkers or the studies that are characterizing the illness, either epiphenomenally or in terms of integral pathophysiology, and in terms of the treatments, whether it's psychosocial or the experimental therapeutic approaches, that you're seeing some encouraging results. Do you feel like you're really producing meaningful results, or it's really kind of incremental results that are painstakingly achieved with conducting studies and pursuing a line of investigation?
Yeah. So I do think there's some areas where we have made substantial progress. So I think one of the areas is in early detection. At Duke, we have a baby clinic. And parents can bring a baby in by 6 months, 6 to 12 months of age. And we cannot diagnose autism at that age, but we do know what symptoms to look for. And we are able to offer parents strategies, parent-implemented interventions that we believe are helpful. And we do know from the studies on early intervention that this does have an impact on long-term outcome. And if you think about the fact that, insurance pays for both autism screening, so the MCHAT you can get, you can bill, right? The APA, or the American Academy of Pediatrics recommends that all children get screened at 18 to 24 months. About 50% of pediatricians screen for autism. Those are pretty, big shifts if you think about actual care out in the community. The majority of states offer an insurance benefit for early intervention. So to me, that's kind of where the rubber hits the road, and I've testified, to the U.S. Senate on behalf of insurance benefits. So I think when you think about that area, And then if you combine it with, for example, the work of Joe Piven, where you can start to see brain changes that are definable at 6 months of age that are early biomarkers for autism, that to me feels like real progress.
Is that the cortical hypertrophy?
That, well, I think what he sees is right between 6 and 12 months of age, he sees differences in the white matter fiber tracts that look like it's actually sort of increased fractional anisotropy early on that's then followed by a slowing. But also with Mark Chen, he's shown this extra-axial CSF fluid, right? So if you have high-risk infants that are being followed with MRI, and if you see this extra-axial fluid, which is quite noticeable, that baby is, I think it's something like 0.8 positive predictive value of going on to develop autism. Now, my colleague Joanne Kurtzberg will tell me that's a nonspecific marker in the sense that you do see that in other neurodevelopmental disorders. But on the other hand, you know, if you think about if you had a baby at risk and you could do an MRI and start to see some of these changes in brain early on, that's, to me, that's pretty exciting. Now, let me just say, though, in the area of drug, you know, which is, I think, the harder one, I think the jury is way still out. Right, we have one, you know, if you look at that vasopressin antagonist that Roche is really aggressively pursuing, it's at the very beginning stages, and I think autism is biologically incredibly complex. And so I feel like that it's a much riskier area. That's why I feel like the things you can control, like measurement, we have to at least get that right so that we can test them, right? So, but on the other hand, I guess the way I think about it is that if you had yourself or a friend who had anxiety and depression, schizophrenia I would say as well, but certainly anxiety and depression, particularly depression now with all the exciting work in ketamine, is that, you know, we actually have things we can do, right? Now, we don't completely understand the biology, but we have something, and what it does is it reduces the symptoms. So I think that is what we're shooting for with autism. It's not that we're going to cure autism, but that we might have things that modulate the brain enough that it actually makes you more, you know, able to interact socially, more able to go out in an environment that's very stimulating and improve quality of life. So I think if we can get there to where we now are with anxiety and depression, I'll feel like that's a home run. But it's still a hard and risky road ahead. And I think, Jeremy, you would probably agree. Yeah.
Well, the early detection is really important because we've shown with the Denver
Early Start Denver model.
You know, if you then implement this very labor intensive, but hands-on treatment, there are improvements, and they appear to be long-term from the data you showed. Other questions? Jeremy.
So great talk, Jerry. I think it's inspiring to think about where we've been and where we are now. And I would agree that early detection, early intervention seems to be making a huge difference. It's hard to measure. It's one of these things where everyone wants to do it. And so it ethically is difficult to go back and say, is this making a difference by comparing to a true control population, at least in this country? I'm curious, in particular, in light of some of the infant sibling work, Sally Ozenoff and others, that really seems to show this step off in social function late in the first year, around 9 months or so. Some of the appeal of the amazing work that you're able to do visualizing behavior and allowing a machine to distinguish things that otherwise previously only expert clinicians could have hunches about. I wonder how early you think we're going to be able to push. I'm actually much less hopeful about the trajectory work that Joe has shown, which is beautiful work, but it's really trajectories over that first year of life that change. I don't see that we can use that work, even if we put every kid in a scanner, which we're not going to do. But that work is really going to lead us to diagnosis. I think your work is a lot closer to being able to do that with fine-grained analysis of behavior. But I'm curious about what you actually think, not where the data lead, but what you actually think in terms of how early we might be able to push.
Yeah, that's a good question. So one thing I'll just mention is, again, when I came back to academia after being at Autism Speaks as Chief Science Officer for 5.5 years, the other thing I made a commitment to is not to do high-risk infant studies. And the reason why is I always try to do things that, I mean, so many people are doing them. There's no reason to jump in and do the same thing. I always try to find something that is different, right? So that, but also I worry a little bit about that most of our knowledge about early symptom presentation is on multiplex families. Right, that's a very specific subpopulation. So every one of those kids is from a multiplex family, and we know that the majority of kids aren't from multiplex families. And also just the selection bias in being in those kinds of populations. So I did make this commitment to try to do population-based studies, and that's why I started working in Duke Primary Care. I'm very lucky at Duke that we have a couple of things that make it easy to do that work, relatively easy. One is that the pediatricians in primary care, and there's many clinics out in the community, are very engaged in research, and it's part of just the culture that they participate in research. And then secondly, with the EPIC system in 2014, we now have a really robust electronic health record that every child is followed with one record from primary care into all the tertiary care clinics, like the clinic that I oversee, and so that you can really track that child, from just being a typical baby that's being seen out in primary care. So I decided to start at 18 months with our measures, partly because that's where we're screening kids now, and we have the MCHAT. So I wanted to compare to what is standard of care. I will say I submitted to an RFA that NIH recently put out on infant screening. If any of you are out there, then we're competing. But anyway, we are going to we did propose to use the same approach at 6 months. In the same primary care sample. So this should be around 9,000 kids in the end, right, that would be seen. And I think that'll test. My hunch is that, I do think Ami's work where Ami Klin showed differences in eye tracking to social attention eye tracking between 2 and 4 months of age. But if you look at that data, it's also a trajectory. Right, it's not really at 2 months you see differences. But what I've seen clinically, right, and also even in that first case study that we published, is that you can, in some infants, not all, because it's quite variable between 6 and 12 and 18 months, but in some infants, there clearly are symptoms by 6 months. I've seen that, right, where, it just, the baby is not paying attention, and they're not babbling, they're not, smiling at you and all of this. So I think I don't know what proportion that is, if that's a very severe phenotype, and whether it's frequent enough that you could pick it up statistically in a large sample. I think these are all questions. So in our study, we did propose to see kids at 6, 9, and 12 months. And, we do suggest that trajectory would be important. But think about it this way. If you started to have concern, and if you had something like an app, can you imagine a physician saying, look, what I want you to do over the next 6 months is once a month, I want you to show this to your baby. And the information is going to be signaled to my EHR record, and it's already going to be quantified by the engineers, and it's going to give me a risk rate. And if I start to see things changing month to month, I'm going to ask you to come back in. That's my vision, right? And I think that is probably possible.
It's like a growth curve.
It's like a growth curve, yeah.
David.
So could you tie up all together in a bundle, screen time, the cerebellum, eye tracking, and it seems ironic that since the CRT screen is the root of all evil for pediatric recommendations, that screen is going to be the solution, that babies are going to be looking at these screens to test it.
Oh, I see, yeah.
But anyway, do you think that the cerebellum eye tracking and looking at the television screen is important?
You mean just in the daily use, what they're doing at home?
Do you think it's ideologically important? Do you think, since we're all moving to look at these screens all the time, and young people don't look at each other, they look at the screens all the time?
Yeah, so,
everybody
well, okay, so
autistic?
the question of, is there some impact in terms of autism of the increased use of screen time? I think that's what you're asking. I actually know that literature because Raphael Bernier, Joel Nigg, and I are just finished a book for parents. It's called What Science Tells Us About Autism, and we reviewed that literature. And so there actually is not strong evidence yet that kids watching screen time is increasing autism. There may be some connection with attention deficit, but not necessarily autism. I do think, however, that one of the weaknesses potentially of the approach that we're using could be that particularly when we get into the game that we're incorporating, which I don't know if it's going to work, but I do think familiarity with digital devices and whether you've ever played a game on a digital device could mess up our results. So we're measuring that. We're asking parents, has your child ever, been exposed? Are they familiar? But even if they aren't familiar, if you show a toddler a little movie on an iPad, they're interested. They watch, right? And so I don't think it's going to be a fatal flaw. But in cerebellum, yes, I think that we know that the cerebellum is involved in autism. It's very much involved in, timing and motor function. We know that motor deficits are some of the earliest signs that we see in autism as well as other disorders. So, I think the cerebellum is important in autism. I think that was.
Did Eric Corshane's original findings?
I think those have held
Has that held
up.
up?
Yeah, not necessarily that everybody has, hypoplasia of all, vermal globules 6 and 7 or whatever it was. But the idea that if you look at the Purkinje cells, I think that has held up on the autopsy, studies.
Bill and then Joseph.
Jeriel, I also echo the inspiring and really impressive story and the work you've done. I'm wondering about if you've been looking at or thinking about looking at related phenotypes. The ones that come to mind are autonomic imbalance, sleep deficits, and GI dysfunction. Are they also going to be part of the outcomes that you're either looking at now or will be looking at with these latest interventions?
Yeah, so in the studies on cord blood, for example, we do have measures of GI and sleep as part of our outcomes. But also in the new proposal that we submitted to NIH to look at early detection during the infant period, there's two pieces of it. One is to look at these digital phenotyping approaches. The other, and this is kind of in vogue, but I think it's really potentially powerful, is the idea of looking to see whether there's elements in the EHR that could help you predict which babies are more likely to go on to develop a neurodevelopmental disorder. So there's a big push right now at Duke. It's called the FORGE Initiative. It's actually headed up by Rob Califf, who was at Duke and then went to be the commissioner of the FDA for a period of time and now is back at Duke. And when he came back to Duke, he established a very big focus on how can we use EHR in a quantitative fashion to be making better decisions for clinical care. So we're already working on this. So we have a retrospective database of about 30,000 children that were seen in primary care from 2014 when EPIC started, which is kind of when you can start doing this kind of work, to now. It ends up like 268 now have a diagnosis of autism. We have certain with ADHD and so forth. And we're already seeing these really interesting patterns. So for example, in the first year of life, you see really significant differences in just clinical care usage, like going to the emergency department. If you're going to have ADHD, which isn't diagnosed to 3, before age 12 months, you've already been to the emergency department more times, right? So in that regard, we are looking to see whether frequent GI or sleep or other problems, medical problems that we know are associated with autism, could show up in the EHR. And the idea is that you would, be automatically just detecting these things coming up in the EHR, and it would be feeding into your algorithm. And then when the physician comes in, they get a little flag, right, that says, look, this particular child has now several risk markers that we know are risk markers for this. And that just means you need to maybe do extra screening, ask extra questions, increase surveillance. So I think that's also a very cool new approach that, holds a lot of promise.
It's like you have a registry.
Yeah.
Joseph?
Excellent talk. I think my question actually very appropriately follows from the previous about kind of phenotypic differences. I was kind of curious what information, if any, you're collecting about comorbid intellectual disability in cases of autism, acknowledging that obviously, autism and intellectual disability are kind of different, right? They're distinct axes, so to speak. And because you demonstrated, for instance, that higher IQ, mediates one of the potential responses to your intervention. So I'm curious, in addition to what information you're collecting about intellectual disability or low IQ, what, if any, you think is the effect of comorbid intellectual disability on some of the measures that you're looking at as potential biomarkers?
Yeah, I think that's a super important question. So the way we're approaching it in that Duke primary care study is any child who gets a positive score on an MCHAT is brought in, and they're comprehensively evaluated, including looking at intellectual disability. So they get an IQ test. And then the children are followed longitudinally, and at age 3, we're screening again. And any child who has either a social deficit on the Vineland or on the SRS, which is another screening tool, at age 3 is also brought in so that we can pick up kids we might have missed, and they're also given an IQ test. And I do think it's going to make a difference, and it could make a difference that could be, a fatal flaw for all we know. We honestly don't know. That's why it's important to have a large population where we'll have kids with, with and without intellectual disability, with and without ADHD. All of these could completely muddy things in a way that could be incredibly complex, right? I mean, I just don't think we know yet.
Well, we're just about out of time, but we're going to have a speed round of a couple additional questions. Lynn's, the oxytocin study, what happened to that? What was the result of that?
Yeah, she is still analyzing it. Yeah, I think the story there is, one, it was a tough transition from UNC. Well, it was a tough study because it got delayed because of not getting the compound. I don't know if you know the story. I won't go into it. It's a long story, but they did not have access, and they had to create it themselves, right? They had to create their own company and create the oxytocin. So that delayed things. And then she had a transition to Duke, which further delayed things. But the data are there. They're cleaned. She has money to analyze it. She's analyzing it. So I think we're all waiting with, bated breath, so to speak, for the results.
What's your guess?
I'm not going to guess if I'm right.
Okay. Secular factors. David asked about screen time, but what about vaccines, paternal age, assisted reproductive strategies, and so forth? Are these contributing to the population frequency, and is there an epidemic of autism?
Yeah, well, I think the data suggest, obviously we can't completely measure it because measurement changes over time. So, we don't have an answer that is foolproof. But I think that both the epidemiological studies, which I won't go through and, but Peter Bierman and others' work who have really tried to parse apart what factors have contributed to the increase, and also our clinical sense is that there is a true increase, right? I don't think it's just better detection. I think a lot of it is better detection. And also, if you look at the epidemiology of autism, it's shifted dramatically in the sense that it used to be in the old days, so to speak, that about 70% had intellectual disability. Now we know 60% do not have intellectual disability, meaning only 40% have intellectual disability. So the population of what we think of as autism is different, which is really important. I keep telling people at Duke this in our adult care. The typical patient presenting with autism is a high-functioning adult who has probably mostly anxiety or depression, right? That is your, I think, prototype adult autism patient. And so I do think things have changed in terms of what autism is, but I also think there's, a number of contributing environmental factors that have changed. Parental age, exposure to pesticides, air pollution, the prematurity, the fact that children now live, who are born very low birth weight that would have not survived. There's a lot of factors. Now, the CDC, thinks that that's a pretty small contribution still. So, I think we still have a lot of questions that need to be addressed.
The DSM change to autism spectrum, how did you feel about that?
I think it was brilliant. Yeah, I really do, because I think what they did in the DSM-5 was they separated, what we think of as autism, which is you have problems with social communication and presence of repetitive behavior and restricted range activities, from all the comorbidities, which could be intellectual disability, regression, your genetic etiology. You could have Fragile X now rather than trying to keep parsing all those off. That would have been an infinite number of conditions. So, you have these two key features, and now you have a lot of other things that explain variability. And I think that was pretty brilliant.
Lastly, what do you think of the relevance of the concept of neurodiversity versus a disorder or disease or syndromal pathologic condition?
My belief is all of the above. Right?
Oh.
No, seriously. I think, as a clinician, the way I approach it is I am here to serve the patient. And if a patient comes into me and they do not view their autism as a disability or disorder, and it's a part of their identity to celebrate, then, that is my viewpoint with that patient. If another patient comes in and has cannot leave the house because of their sensory processing problems and has seizures and GI and wants to just get rid of autism and couldn't it be cured, I respect that too, right? So I feel like it's really not my place to make that judgment. I honor those different perspectives and I work with those perspectives. Just like I almost think of it as when we get cultural training. Right? We do have to work, and not have to, but we want to work with our patients where we respect the cultural differences that people bring. And, I've had parents who come in and say, his hand flapping, like the world should celebrate that. Why can't we hand flap, for gosh sakes? And then I remember working with a family who was Chinese, and I was saying, I think it would be good if you didn't pressure him around this issue of hand flapping. And she said, in my culture, being the same is really important to me. And so, it's not for me to judge.
Well, Jerry, thank you for a wonderful talk and for all the wonderful work you're doing. Thank you.
Thanks.
Unfortunately, there's not going to be a lunch as usual because the multipurpose room is undergoing renovation, but Gerald, stick around for a couple of minutes if you have any questions.
Well, that was fun. It was fun to see you and get to have a little fun exchange. I didn't know you.